All right, up next, we've got Kellen Nelson. And Kellen, a research forester for the USDA Forest Service Pacific Northwest Research Station in Juneau, Alaska. He received his BS and MS in forestry from Colorado State University and a PhD in ecology from the University of Wyoming. Kellen's research investigates how climate change impacts forests and forest disturbance regimes, and how silviculture can be used to improve stand conditions and deliver ecosystem services. And today, Kellen's going to talk about the Congress-wide young growth study, among other things.
So if you're loud, you don't need to use the mic because we can hear you in the room, but the owl is what is the mic for Zoom.
All right. Well, I'll turn this off. You know, I looked at what I wrote in the description for my abstract, and man, I was aspirational. I was going to hit a lot of different topics, and I prepared this presentation. I think for the 30-minute time slot, we're really going to focus in today on patterns of deer browse across gradients of slash and forage. And this is a big hot button issue, I think, across all, you know, not just the Tongass and my national forest partners, but also state and tribal folks as well. Certainly the Mule Deer Foundation is going to be paying attention, and we are just getting started. So I noticed that we just dropped my presentation in the chat here. Please don't take that and share that around too widely. All the analyses that I'm going to present today were conducted in about the last 3 weeks. So we are really like hitting the ground running and accelerating. So I'm really excited to present to you. So I'm presenting here, but this also shows work by Phil Manlick, research wildlife biologist for PNW Station, Jeff Barnard, who he's been a field biologist and has led the Twigs crews since the inception of the project, and really has been the backbone of the data collection here. We have a lot, we owe him a lot of gratitude for keeping this data set together and this program going. Jen Wen, our new data scientist, is here today, so take the opportunity to talk forestry with her and what you might be interested out of this data set. And then Mike Howe, who was mentioned earlier, did some analysis. So I'm going to just thank, I mean, long-term monitoring projects really take long-term funding, and you know, Region 10 and the Tongass has put forward that funding for over 20 years to keep this going on. So, and then I'd like to acknowledge Mike McClellan, who many of you know, and Justin Crato, who is now in Montana, but held the reins for a year and has gotten some worthwhile products out as well. SCA, Student Conservation Association, has been a partner in all of this work. All of our crew members were SCA. We've had over 150 people, and we have people from all that are in all different corners of natural resources now. By my estimate, maybe about 10% of them have stayed in Alaska. So I think that's a success of the project in itself. And I keep hearing back, I keep learning about new faces of people that participated in the program. So that's been great. So a little bit of what I'm going to talk about today. We're going to talk about the setting. We're going to talk about second growth forests on the Tongass National Forest. We're going to talk briefly about even-age forest dynamics. I know this group is really familiar with that, but we're just going to review a little bit. We're going to talk about the study for twigs, and then we're going to dive in and we're going to talk about these recent results on patterns of deer forage across gradients of slash and forage. So I think a lot of folks in Southeast Alaska are familiar with this. This graphic produced by Alan Brackley in 2009. On the y-axis, it shows million board feet, and it shows our harvest rates through time with different ownerships. You can see in the 1950s, really things ramped up across the Tongass and across Southeast Alaska, and they really ramped down about 2000. And, you know, so this gives us a nice time frame on how much was produced. You'll notice that the tribal and private lands really ramped up in 1980, following Anilka, and they also kind of ramped down in the early 2000s. So Brian Kleinhans and Terra Verde, through a number of partnerships, went through, and they've done a great mapping job recently. And so I pulled a few figures out of their report, and I just wanted to show off kind of what is young growth in Southeast Alaska. And really, we're looking at these stands that were cut since, over the last century, but predominantly since the 1950s. On the left, you can see annual acres harvested. We have 5, 10, and 15,000 acres per year. So you can see we had years over 10,000 acres a year in the late 60s and early to mid 70s, and then again in the late 80s to early 90s. And then on the right, you can see cumulative acres harvested. You can really see that rate ramp up quick in the 50s, and then it levels off again after 2000. So all told, according to Brian's report, and there's other data sets as well that put us about in the same range. We have about 450,000 acres of young growth on the landscape. We estimate about 42%, or Terra Verde estimates about 42% of those are going to be removed from the young growth base. I'm sure that that figure could differ a little bit, but what that comes to is about 260,000 acres of net young growth in the timber base that we can manage moving forward. So when we take that same data and we look at the age classes moving forward, you can see we have this light, we have age classes, we have this light, tan color on the left. And you can see we have a whole lot of stands that are under 30 years old. This thinning window is kind of ideal window for pre-commercial thinning. And it depends on site productivity, but I had to put some brakes on this graph. So this is what we came with. And then in green, we have 30 to 60 years, which is kind of post pre-commercial thinning, but before commercial thinning. Then we have the 60, 90 degree window in teal, and we have 90 to 120, which is approximately a rotation age, and then over 120. But when we look back, when Twigs was designed in the 2000s, if you look at that far left bar, you can see there was a ton of acres for pre-commercial thinning. This was a real major issue, and we established this monitoring program that I'm talking about. In the 2020s, and I'm not sure if I have my bar in the exact right place, but you can see that this pre-commercial thinning window is narrowing, and we have fewer and fewer acres in pre-commercial thinning. Now, when we move forward in the 2040s, there's not going to be nearly as many acres. Sure, we're still cutting, and we're going to produce some more acres that might reach pre-commercial thinning. But what we've cut today, there's not going to be a lot in the 2040s available for thinning. But what are we doing in our pre-commercial thinning? We're trying to set up the stands for future management, right? We're trying to provide ecosystem services that are sort of lacking this early phase of stand development. And we're trying to set those stands up to be more wind firm, to have more management options. So if we want to get in and do commercial thinning, we want to do restoration activities, we want to do something in those stands, they're more resilient to disturbance. So we're thinking forward, we're thinking 20, 30, 40 years forward, and we're thinking about what is now a young age class will be an old age class, and we're trying to set those things up. So just to review forest developmental model, this is from a Nowacki and Cramer, 1998. This is a wind GTR, but it illustrates things pretty well. So we're really focused in this first 100 years where I have the orange box, but we have stand initiation. This is shortly after cut. The trees are coming in. You can see it's relatively open. We have a lot of forage. We have the stem exclusion phase, which is really dense. Those trees are competing with one another, so we're getting mortality. We have a closed canopy that is preventing light from going down to the forest floor, and we don't have a lot of deer forage in there. It's pretty visible in the example that I put up on there. In the upper right, we have understory reinitiation, and that's when the canopy starts to break down after the stem exclusion phase. I show a canopy gap in this sense, or in this case, and then forest in the lower right. So Paul Alaback in 1982 had this great paper where he really just shows understory biomass in this relationship here, where you have your stand initiation phase. We have a lot of understory biomass when the canopy is open, and then it just tanks. As you enter that stem exclusion phase, we don't have a lot of light hitting the ground, and we have far less understory biomass, and this translates into less forage for deer. So about the same time, all these folks were really discussing and working together on these issues. We had anyway, I'm not sure of these folks' first names, but this document Wallow and Show in 1980 has this great figure where in the upper part of the graph, we have deer relative carrying capacity, and you can see that we harvest once at the beginning, time 0. During stand initiation, deer relative carrying capacity increases as we're, recovering that stand. And then you enter that stem exclusion phase and the relative carrying capacity decreases. The lower part of this graph depicts how maybe a rotational approach would take, so that whenever you enter that stand initiation phase, you get a bump up in the relative carrying forage, and then it declines through time and you have low amounts through time. So that is sort of the context for twigs, and we're interested in doing pre-commercial thinning to, attenuate and add back some of these ecosystem services. So our questions for this study, I didn't have time to make a nice graphic, so I'm just going to read through. And these are our assumptions for what we're talking about. We're looking for the ability of pre-commercial thinning to improve deer utilization, and they're going to do that through accessing forage. So our first question, how does deer browse vary with slash and forage quantity? It's a really basic question, right? Our first hypothesis, deer browse declines with slash cover and depth. Second hypothesis, deer browse rises with forage availability. We're all pretty familiar with this. Our second question, how does stand treatment age influence browse, slash, and forage? So if we take that naturally regenerated stand and we move forward in our successional sequence, what happens when we intervene with pre-commercial thinning at different points along that successional sequence? So we anticipate that deer browse will decline with stand treatment age as those trees get bigger. We anticipate, or we hypothesize, I should say, that size with stand treatment age as the material gets bigger as we thin. And then our third hypothesis, forage declines or is neutral with stand treatment age following pre-commercial thinning. And then our third question, how does deer browse, slash, and forage vary with time sense treatment? And so, forests are dynamic. I always like to make that point. I think that's the thing that I always want to really reinforce to folks, that these things vary through time. And we go in there, we might do an intervention or we might leave the stand alone, but nothing is ever the same in a decade, in 50 years. Stands are all different. And so we expect deer browse to rise with time sense treatment. Second hypothesis, we expect slash to decline due to time sense treatment due to decomposition and compaction. And we expect forage to rise as it recovers from pre-commercial treatment. All right, so the twig study design. This is the great design. This thing is terrifically replicated. We have data going all the way back to 2007, 3 measurement cycles. So we have 3 experiments. We have experiment 2, 3, and 4. So experiment 2, we went in and did pre-commercial thinning at 15 to 25 years. 3, we went and did pre-commercial thinning at 25 to 35 years. And twigs 4, we did pre-commercial thinning at over 35 years. So today we're just going to be talking about thinned versus unthinned. That's the control I'm referring to as a control. There's also some other treatments in here where we did some pruning, we did some girdling and some bucking. I'm not going to talk about those today. Those are going to be future analysis. We're just going to talk about thinned and control for now. So these stands, these treatments were implemented 2002, 2003, 2006 for twigs 4. And you can see the measurement years where we got back in there, we got the crews out, and we went and remeasured these sites. So this is probably from 2 years ago, almost 20 years after the thinning. And you can see on the left, we have the controls. And so we have twigs 2, which is now 30 years old. It's still pretty darn dense. There's a lot of trees that are dying in there. It's closed canopy. And you can see this progression from top to bottom as that stand ages, it gets sparser and sparser, right? And those trees have come down and passed away and fallen on the ground. On the right, we have the treated stands. And I really wish I had been in those sites in July when there was a ton of forage, but I was in those sites at the beginning of June. I think you can get the sense that there's a little bit more understory vegetation in there. You can see some bushiness in the bottom, but you can see that the canopy is open. The trees are more spaced out, and we do have more understory veg. So if I was in there in July, we'd have, chest deep vegetation in a lot of cases. And I just wasn't there to take a picture at the right time. So on the right, we have a map of where these things are distributed. We're particularly interested in Twigs 2 is in red, that's the youngest one. Twigs 3 is in yellow, and Twigs 4 is in orange. And so you can see that we have a lot of sites down in the Ketchikan, Prince of Wales area, but we also have sites around Wrangell, Petersburg, and farther north than that. So we have pretty good, as far as our logistics are concerned, you know, what we could permit. We were able to access a broad distribution across the landscape. All the layouts for these treatments were different. The treatments were generally, I think, between 8 and about 20 acres each. And so the depiction on the right, I'm sorry, or on the left, sorry I didn't redraw this with a little bit larger font sizes, but you can get the sense that we have different layouts with different treatments. We have thin treatments, we have, and then we have those specialty treatments. In each of the treatments, we put approximately 5 plots. And so we're not advancing what I do. All right, so here's our plot design. It's a 5th acre plot design. And in the center, we put a cross with these 1 by 1 meter quadrats. And so the data that I'm presenting today, we are really focused in on these 1 by 1 meter quadrats. And so we put 60 quadrats out per site, 5 plots. So generally we had 12 quadrats per plot. So inside of these quadrats, we measured the slash. We measured the percent cover and the depth of the slash. We measured the understory plants, so species, percent cover, tree ingrowth. And then we marked whether the deer browse, yes or no. So this is really terrific data, right? We know that the deer went to this site. We know that the deer ate plants right there, and we know the plant community that they're eating out of and how much slash. So we know that the deer access those locations. So we're going to start off, I'm going to walk through the plant community data first. It's not the community data, but really it's the forage quantity data. We're going to walk through the slash data second, and then we're going to look at how plant communities and slash predict deer browse rates. And the results are really cool. So I'm going to hit you with some data, and just beware, you're going to be kind of overwhelmed here. There's a lot of points on these graphs. All right, so all these graphs look approximately the same. And we have 4 windows, right? On the left side, we have Twigs 2. That's when the pre-commercial thinning happened at 15 to 25 years. When in the middle, we have Twigs 3. That's 25 to 35 years when the thinning was implemented. And on the right, we have Twigs 4. That's greater than 35 years when the thinning was implemented. And then we look at the x-axis. The x-axis is time since treatment. So these are our remeasurement times, and we went back. And so this first one, we're starting out, we're looking at dry biomass. So we took all those plant species, we took the percent cover, and we were able to estimate how much biomass each of those plants produced. And we summed them all up for every quad and every plot. So every dot that you see there is a plot that was measured in time. We're often used to seeing averages, right, with like error bars. And so I could turn this data into averages with error bars, but you're not going to see the variation here. And I think it's really that we have these conceptualizations of what happens when thinning occurs and at what age you do it. And it turns out when you measure a whole lot of plots, you see a huge diversity in what you get out of the other end. And I think that that's really going to be important, especially when we get to the slash data here. So what do we see? In ink blue, we have the controls. And then in the colors, we have the thinned. So on the far left, we thinned at 2 densities. The light blue is the denser of the 2 thinning densities. And the the light green is the denser thinning density. And the dark green is the sparser thinning density. And so you can see this progression of producing more forage depending on your thinning density. You increase the canopy more, you get more forage produced. Same goes for all the other treatments. So one thing that's really interesting when I started looking at this data, I expected to see an increasing progression in these data and these plots. So you see the box there, the upper and lower bounds of the box. The lower bounds is the 25th percentile of data, and the upper bound of the box is the 75th percentile of data. That line in the middle is the median. It's the 50th percentile data point. I expect these things increase with time, but they stay pretty darn steady. I mean, maybe we can conjure, there's a little bit of an increase through time, or it goes down, it goes back up. But what this tells me is there's a huge response that happens within the first 5 years. We started measuring these things in the first 5 years. We get a lot of understory recovery in that first 5 years. And I thought that was really unique. That was something that popped out to me. But, you know, our assumptions on thinning and producing understory vegetation when you thin the canopy, they're pretty much upheld. So we took that understory biomass and we went back to Tom Hanley's work and all the GTRs he published, and we got into the fresh model, and we took biomass, and there's conversion factors from every species, and we converted biomass to digestible dry matter, and then from digestible dry matter, we're able to convert to digestible protein. And so this is summing up the high quality forage in all of these sites. And the reason we do digestible protein, I'm presenting it to you, is because when we got to that modeling at the end that I'm going to show, it was the most significant thing that predicted deer browse rates. So that's why I'm showing you both the biomass and then this derivative digestible protein. And we see the same, right? So we see more digestible protein produced in the thin stands than in the control stands.
No idea what's going on, but I can't hear you.
All right, I'll use the mic. We have, you know, the garbage truck or something came, so such is life. But the same pattern here, you know, this largely reflects the biomass data. But again, I just wanted to show that these are the units that we're really interested in. Digestible protein, very great for deer. All right, patterns of slash cover and depth. So here are patterns of slash cover. We went in, we marked the cover, percent cover of slash. You can see that we have in twigs 2, a relatively sharp decline through time, where we produced almost 50% slash cover, almost 50% of the ground was covered in slash. It's more than half now, 16 years, between 5 and 16 years. You can see in twigs 3 in the center, when we produce kind of that when we thinned that medium size wood material, the decline is not quite at the same rate. Now, when you look at the right, wow, it's almost pretty stable, right? If you thin a bunch of large trees over 35 years old, well, the slash just doesn't break down, doesn't decline as quickly. So we're going to dive in here. Now on the y-axis, we have slash depth. And I think slash depth is great because it's something that we can actually design a prescription about. We could tell a contractor, hey, don't exceed this slash depth. Unfortunately, we didn't use it in the model at the end because we're missing this data point. You can see it in the 10-year mark right there. It's when we went from paper data sheets to using the Allegros, and someone didn't program it into the Allegros. So we're missing a whole year's worth of data there. We're going to find years to really get back into that and come up, because this is important, right? It's like you can write a prescription with slash depth. So we're going to get in and explore this a little more, but it's not used in the model at the end. But I just wanted to show the trends here, that we have a similar trend. We have the declining slash depth. We get compaction and decomposition occurring. All right, so this is great. Now let's get into the modeling. So we took the slash variables, we took the forage variables, we put them in a model to predict whether or not, this is a yes or no question, a deer went to that quadrat and measured it. So over the course of the study, we had about 15,000 observations of these quadrats. So data, right? 15,000 observations. And of course, they're split up with all the different treatments and, you know, time sense treatment, but there's a lot of strength in these models. So patterns of deer browse with treatment. It's really clear that thinned forests have far higher rates of deer browse than unthinned forests. And what stands out to me in this graph is the light green color. This is twigs 2. This is the stuff that was thinned between 15 and 25 years. You have a really big effect size. And I put that bracket there to really show like how much of the effect size is so great. Twigs 3 in yellow, that was thinned between 15 or 25 and 35. Also, I mean, obviously a substantial increase in deer browse in thinned over unthinned, but less of an effect, less of a magnitude of effect. And in twigs 4, the ones that were thinned over 35 years, and we put some big material on the ground, Still positive, but it's far less of an effect than thinning early. So yeah, thin early over thin late. I think that that's pretty clear in the data. All right, so how does time since treatment work?
What was that? Can you go back? What was that second point? Decline with treatment, what?
Or did I not finish my thought in the bullet? Declines with treatment.
It's like you just kind of the Zoom screen.
Yeah, but I don't know that there's another, oh, treatment age, I see. Yeah, so treatment age, so it progresses right from green to yellow to brown. Yeah, thanks, I'm gonna, drag these things all around. Don't want to put on Phil's name. All right. Hit forward a couple times, let's see. It's when I touch that thing, all right. All right, here we are. So now we have browse probability again on the y-axis, and we have time since treatment. You can see these treatments really clearly differentiate here. We have a large magnitude on the left in twigs 2. We have a narrow magnitude on the right, twigs 3. And the change through time, there's actually a subtle, the slope on that upper thin line is a little bit more pronounced than the slope on, on twigs 2 has a greater slope than twigs 4, let's just say. And you can see the difference between the treatments narrowing, but it's positive. We're seeing an increase in the rate of browse through with treatment and without treatment for that matter. All right, so browse probability, here we are, digestible protein. This is interesting. So we obviously get a lot of differentiation. And I got to say, there's a lot of stands that are falling on the left side of this graph that are driving this model. So we get a lot of differentiation. The thin stands are producing a lot more digestible protein. There's a lot more deer browse in there. But as you provide the protein, the deer are coming to it, whether it's in the control stand or if it's in the thin stand. And so if you provide the groceries, they're gonna come feed off of the groceries. And I think that that's the moral of the story from this graph. Yeah, so again, thinning early over thinning late. All right, so here's the slash cover one. And we have percent slash cover 0 to, it's not percent in this case, it's a proportion. Didn't quite get that axis right for you, but we get the picture, right? So yeah, as slash increases, we get a decrease in browse probability. There's a major difference between the treated and the untreated on the far left, and it narrows through time. Yeah, so treatment age, we're producing a lot larger slash, and we hope to quantify that a little better in future research. In the right on twigs 4, and we're seeing that it takes a toll on the rate of deer browse in those treated stands. But hey, we're still getting a lot more deer browse in the thin stand, even with the slash, than we do in the control stand. All right, so I'm going to conclude there. Just remark that our assumptions of greater forage and greater slash in thinned over unthinned stands are supported. These are the hypotheses that we were talking about earlier. They're very general hypotheses, but it's nice to see that they stand up in the data. Forage responds rapidly within 5 years following thinning, and then any increases after that are minimal. Slash declines most rapidly when thinning material is small, when you do the thinning early, and deer browse declines with slash and rises with high forage quality. Yeah, so again, deer browse response is greatest when stands are thinned early. And I think the biggest thing that comes out of this is thinning is a huge driver or strong driver of deer browse, and greatly outweighs the negative effect of producing the slash itself. You might be able to go in there and optimize a little bit, but it's pretty clear that the deer get in there and get into the slash. So I'd love to take some questions. I'm not sure if there's time, but maybe a couple minutes.
Yeah, I was just wondering, it seems like with browse and pioneer browse, winter versus summer, and how Hanley's work with digestible protein was from research in the 70s or something in the winter, with lichens coming out of the trees and I mean, there's an interplay between the timing of forage production and digestible proteins and what's important at what time of the year things. Deer, to try to equate deer survival or persistence in our changing climate with summertime browse seems, anyway, thoughts.
Can you repeat the general concept of that, Christian? Is that online audience?
Yeah, so we got a question in the audience that was, I think, asking about, and you can clarify, asking about summer versus winter utilization of the forage and the browse by the deer. And Tom Hanley did a lot of work looking at lichens and looking at wintertime deer browse, in addition to summertime deer utilization. So, what was the question?
Well, it's how we might interpret results from summertime response and forage, primarily in the understory, with, ways of managing that might focus more on winter time.
So I think, yeah, I think I'm getting where you're going from. So how can we manage to optimize summer and winter, or summer versus winter? And I think that that's kind of where we're going here. And, there's a lot of work to come with the plant community data that we have. In some cases, I'm really interested to see how these different species change through time. In the winter, the deer are utilizing, the blueberries, they're eating the twigs, they're getting at Cornus canadensis. If there's not a lot of snow cover, there's some evergreens, that sort of thing. My colleague Phil Manlick has been picking up deer pellets, or he hasn't been, he's been having people pick up deer pellets all over, and they can take the chlorophyll DNA from those deer pellets and identify to the species. So there's going to be some research coming out on that, and they've been collecting pellets all through the season. And so it's really cool. You get to see the skunk cabbage, like, is the predominant piece of the diet in the spring, right? And then you move into the blueberries and some of the grasses during the summer. And then as you move into the fall. So I think, I don't have the answer for you at this time, but I think diving into both the plant community data as part of this to see how the species respond and change, and maybe classifying those species with summer versus winter forage functional traits, for lack of a better term. And then also looking at that pellet data to see definitively what the heck are the deer are eating. I mean, you look at the chlorophyll data and you can tell to the species what they're eating. Does that get close?
Oh, that's cool. They will tell exactly
Yeah,
what they're eating and when,
sure. I find it amazing. Chris.
Helen, first of all, thank you. This is really great. If I was to summarize this up, and I'll take away to brain and later read some more over this, but PCT, great for deer. Woody residues / treatment, even better, but not necessarily required in every acre. Did we say that?
I think that that's what I'm coming to, yeah. And I've been thinking about what, the trade-offs here. With this data, I hope to be able to do an analysis. If we had prices on like / treatment versus acres treated in PCT, we could do kind of a trade-off of like, how much does it cost to treat an acre simply with PCT with no / management? And then to add that / component on there, what are we getting from managing our / and how much does it cost? And if you're like, you work with private land, so if you have, a relatively small extent and you have money available to do a / treatment, you're going to get the deer in there a little bit more quickly when you manage that /, right? And if you have a high value area that for whatever reason, you just really want to have available to the deer, you might want to do some / treatment. But I think on the other hand, if we're just thinking about producing the most habitat as possible, and there's other areas where we might want to do that, right? My gut, and I have not done the analysis yet, is that you're going to get more bang from your buck across more acres by not necessarily doing the / treatment.
You make your PCT crews very happy also.
Go ahead, Sheila.
I was here when we implemented Twigs 4, and you haven't looked at the data on the / treatments, but we ended up paying in 2006. For example, we would do regular pre-commercial thinning, and it costs, anywhere from like $350 an acre But when we added the slash component, you know, and part of it was to look at the cost and it, we paid, up to $1,200 an acre for that slash treatment. And then we actually had a couple defaults, because they got in with their head, and then one contract defaulted, and we asked a contractor in the area if they would take over that contract, name and price, and they said no. Right, so that was a really big factor, but then also, and I don't know if you looked at that data yet, like the initial put slash on the ground actually impeded the understory. And that showed at 5 years after. I'm not sure if you've been able to look at that.
We haven't dug into the bucking data. So the treatments Sheila's talking about, they bucked the logging slash to 5-foot and 15-foot segments onto the ground. And so, you know, in walking through those stands, you can tell that the slash depth is substantially less than the 5-foot because it's like more compacted, but it's definitely spread more evenly over the ground. And so, yeah, it'd be fun to sort of brainstorm what to look for in that data. I'll go over here, sorry.
Sheila, did you have price differences for when you applied the bucking relative to just a?
I think she said 1,200 an acre.
Yeah, you know, it went from like $350 an acre just from the straight thinning to like 1,200, you know, 1,500 an acre to treat that. But this was like big. It was also these older stands. But we have done some slash treatment since then, but it is a lot more costly. And we do find it hard time, you know, contractors don't prefer that work.
The deer also built like long pieces with all slashed up, so you.
I don't know if that's true. I mean, there's deer in all of these stands. It's like we crawl into some of these stands and then every skunk cabbage is totally mowed down and there's pellets everywhere, right? And it's like, how the heck did the deer get in there?
This is really cool because I worked with Tom Henley, right, you know, especially right before he retired. And he really, you know, he admitted that his thinking on thinning kind of changed over time. But I remember distinctly one of the last things, you know, he was in a stand, it was so full of slash and he's just goes, but doing any is better than nothing. And I think your data is really, really showing.
Yeah, I'd love to talk more about all of this. I think I'm really excited about what we have to do. I think we outlined about 3 papers out of this one set of understory data already, but I don't know that we'll get all the way there. But if anyone wants to talk later, I want to respect the next speaker, but let's talk more about this because I'm excited about it.
Is that all right if I ask one question from online? Because I won't be able to talk to you later.
Yeah, please, Nathan, go ahead.
Yeah, just a question. I was looking at like maybe slide 25 or 26, and it looks like the old stands compared to the young stands for the deer quality, or sorry, the deer browse probability. It looks like those older stands are like half of what the 15-year-old stands or the young stands when they were thinned. But if I remember right, the forage quality was not all that different between the older thin stands and the younger thin stands. And do you have any speculation why that you might see that such a big difference, why the deer are using the young stands so much more than the older stands?
My guess is that, you know, looking at this, I think that there's a little bit more forage availability, although I do see your observation. I'm a little shaky there, but I think in some cases it's due to the slash. I mean, my gut is that there's a penalty to putting that large slash on the ground, and seems to decompose more quickly and the deer get in there more quickly when the slash is small diameter and smaller. But I'd love to follow up with you, Nathan, about that. So I don't have my slides numbered. So you mentioned slide numbers. I don't know if you can see that online, but I'm not sure which ones.
Oh, I was using the numbers on the PowerPoint we downloaded. It gives a page number.
Oh, right on. Nice. All right.
All right, well, I think we probably should, in the interest of time, wrap up here with Kellen. And you're around through tomorrow, is that right, Kellen?
Yeah, I'll be here on departing tomorrow night, so.
Very interesting, great presentation. Let's give it.